Thank you for attending my talk. I am Zahra Adahman, a PhD student in Dr. Carcea's lab at Rutgers University in Newark, New Jersey. And today I'm going to be giving you a brief run-through on our project, Neural Circuits for Social Behavior Adaptations to Environmental Temperature. The perception of changes in environmental temperature is essential for our survival, and the thermal information is processed by the nervous system. Temperature affects social behaviors in different ways. For example, at the top here, in cold temperature, more affiliative behaviors such as huddling, cuddling are displayed. For example, in rodents and in penguins, more huddling behaviors are expressed in cold temperature. While at the bottom here, in warm temperature, more agonistic type behaviors such as dispersion, avoidance are displayed, and in human studies have shown that there's an increase in crime rate in the warm summer months compared to the colder months. Temperature is critical for several social behaviors. However, for this project, we focus on the maternal form of social behavior. And studies have shown that temperature is essential for this maternal behavior because of the neonates, the pups. Pups are hypothermic, meaning they are unable to maintain their internal temperature. This study looked at the internal temperature of pups at different ages when they are separated from the nest compared to a dead pup. And what they noticed was that their internal temperature drops drastically after they are separated from the dam. Until around 18 days of age, they are able to maintain their internal temperature. Another study looked at how temperature is important for maternal behavior by providing a warm nest area with an artificial heat supply and compared it to room temperature nest area. And what they observed was the dam spends less time when you provide a warm nest area. This shows that maternal behavior involves providing warmth for the pups. To understand how environmental temperature affects maternal behavior, we decided to conduct a cohousing assay that is modeled after the nuclear colony formed by mice in the wild, where we cohouse a dam and a young female, a virgin, along with her pups. And what we observed when we looked at the variable of nest occupancy, we tested this behavior at room temperature, thermoregulatory, and cold temperature. What we observed was that the dam and the virgin spent significantly more time together in the nest at cold temperature and spent significantly the least time together with the nest empty. When we looked at the change in the nest temperature compared to the beginning and the end, what we observed is that the animals in the cold temperature have a higher change in the nest temperature compared to the other two conditions. We also conducted nest building behavior assay to look at the effects of changes of environmental temperature in maternal behavior context. We did this by placing the dam along with her pups and a white square unused nestlet material, as you see here, and we leave the animal for 4 hours with enough food and water. And at the end of the recording, we take images of the nest built and we score it in a subjective manner. We designed this scoring rubric to be able to quantify that. And here, as you see, what we observed is that at cold temperature, the animals build the highest quality of nest compared to cold and compared to room temperature and thermoneutrality. We also looked at the area and the perimeter, and what we observed is that cold temperature built a significantly larger nest in area and perimeter. Changes in environmental temperature is processed by the nervous system, and the circuitry begins from thermoreceptors of sensory neurons that innervate the skin and the spinal cord. The thermal information is transmitted via the spinothalamic tract to the somatosensory cortex in the brain for thermosensation and to the perioptic area for thermoregulation. To understand how the circuitry of social behaviors innervates those of thermoregulation and thermosensation, we're going to look at what neurotransmitters play a role in both. And what we found from literature research was that oxytocin and vasopressin, which are involved in social behaviors, specifically in maternal behavior, are involved in thermoregulatory processes as well. For example, vasopressin has been shown to impair nest-building behavior by several studies, and oxytocin have been shown to enable cold defense behavior. Oxytocin and vasopressin are neuropeptides produced by specialized neurons in the paraventricular nuclei, the PVN, and the SON supraoptic nuclei of the hypothalamus. Studies have shown that there are specialized neurons that project to the PVN from the perioptic area. Specifically, they are cold responsive neurons in the median perioptic area that project to the PVN, and warm responsive neurons in the ventromedial perioptic area that project to the PVN. Hence, there is a gap in knowledge about the connections between the perioptic area, the thermoregulatory center, and the PVN, which has prosocial. Hence, there is a gap in knowledge about the connections between the perioptic area, the thermoregulatory center, based on our wild-type behavior results and our literature research, we decided to focus on the vasopinergic aspect to the circuitry involved in temperature-dependent social behaviors. And these studies here are examples of why. In this study on the left, they performed nest-building assay in monogamous mice, and they exogenously supplied oxytocin and vasopressin. And what they observed was that vasopressin reduces nest building quality, reduces nest building behavior in both the mothers and the fathers, while oxytocin did not. And on this study on the right, they looked at vertebrate rats that naturally lack vasopressin expression, and what they observed is that there's enhanced huddling behavior in both males and female in the homozygous. Taken together, this shows that vasopressin play a role in temperature-dependent states by probably reducing more affiliative behaviors and maybe enhancing more agonistic behaviors. To map out the circuitry for temperature-dependent social behavior states, we use the TRAP2 transgenic animal, which is induced to express TD tomato reporter in activated neurons to temperature changes. For the assay, we expose the animal to 35 degrees temperature for 6 hours after it's been injected with 4-hydroxyl tamoxifen. And then it was returned back to room temperature condition and allowed for a week to express the TD tomato endogenously. Then we exposed the animal to the warm temperature again, and immediately we perfused and stained for C4. And we're able to confirm previously shown results that warm responsive neurons are located in the ventromedial perioptic area. We also looked at these trap animals and stained for vasopressin neurons, and we see colocalization within the PVN. Here I have it highlighted with a yellow box around. So we see colocalization of PVN showing that they are warm temperature sensitive neurons that are vasopressin neurons within the PVN. We also looked at the SON, and we see that we had colocalization within. In summary, we observed that temperature influences nest occupancy, the change in nest temperature, the area and the perimeter of the nest built in a social context. We also see that the suppressor neurons in the PVN and SON are activated by warm temperature, and we confirmed that warm thermosensitive neurons are located in the ventromedial periodic area. For future directions, we plan to perform retrograde and anterograde tracing. We plan to map regions activated by room temperature and cold temperature in the trap animal. We plan to perform optogenetic and chemogenetic manipulations, as well as perform single unit recordings of the suppressing neurons in response to changes in environmental temperature. And on that note, I would like to thank the graduate school, my advisor, Dr. Carcea, my lab members, Rumi, Hunter, and I would like to also thank our lovely collaborators. Thank you.